Hello, this is Dr. Ross, and this is the video lecture on the eyes and vision. It's from my class as well as Dr. Camp's Anatomy and Physiology at Lee College. Okay, so just a quick introduction for vision. So we rely more on vision than really we rely on any other special sense. Basically, our eyes are elaborate structures containing visual receptors that enable us to not only detect light, but also to create detailed visual images. And we're going to discuss two general areas of the eye, the internal eye structures, which are going to contribute to vision. And we're going to begin this presentation discussing accessory eye structures, which provide protection. Okay, so the first accessory structure I want to talk about are the eyelids. They are called the palpebrae, if we're talking about them in plural. They're a continuation of the skin, and the continued blinking of your eyelids keeps the surface of the eye lubricated. They kind of act like windshield wipers, so they remove dust and debris, and then of course they will also protect your eye surface when they close. So you have the upper eyelid, which is the superior palpebra, and the inferior eyelid, which is the lower eyelid, which is the inferior palpebra. The next structure I want to discuss are the eyelashes. The eyelashes are there to prevent foreign material from entering the eye. So the eyelashes themselves are very robust hairs, again, and so they're going to help prevent things like dust or debris or even insects from reaching the surface of the eye. The eyelashes are associated with sebaceous glands, and so along the inner margin, as you can see here in this image, there are these modified sebaceous glands called tarsal glands, or meibomian glands, and they secrete this lipid-rich substance that keeps the eyelids from sticking together. Now, you can get an infection or a blockage of these glands, and they result in this small lump or a cyst in this gland, and that is what we would refer to as a stye. So the next thing I want to discuss is the epithelium that actually covers the inner surface of the eyelids as well as the outer surface of the eyeball itself. So that is called the conjunctiva. So you've got the conjunctiva that covers the inner surface of the eyelid. That's called the palpebral conjunctival. And then you have the bulbar conjunctiva or bulbar conjunctiva, or ocular conjunctiva is sometimes called, that covers the anterior surface of the eye. And so it's basically a mucous membrane. There's mucous cells that assist the accessory glands in lubricating the surface of the conjunctiva and to prevent drying out as well as friction. You can get infection in the conjunctiva called conjunctivitis, and this is also referred to as pink eye. It gets this pink color due to the fact that the blood vessels have dilated, making them much more visible than usual. The next accessory structure I want to discuss is the lacrimal apparatus. So the lacrimal apparatus produces tears, and they keep the conjunctival surface moist and clean. So tears reduce friction, they remove debris, they prevent bacterial infection, and they provide nutrients and oxygen to the conjunctival epithelium. Tears themselves are slightly alkaline. That means their pH is slightly higher than 7, and they contain an enzyme called lysozyme. And so these properties help them to prevent infection. So lysozyme actually disrupts the cell wall of many types of bacteria. Now, the lacrimal apparatus itself, like I said, produces tears. It also distributes the tears and removes the tears as well. So each eye has its own lacrimal apparatus consisting of the lacrimal gland. Okay, so this is there to produce about 1 milliliter of tears each day. And then the tears drain into the lacrimal canaliculi, and that's located near the lacrimal caruncle, which is kind of that crease right there, that little pinkish area right at the inner corner of your eye. So these paired lacrimal canaliculi are going to drain into the lacrimal sac. And the lacrimal sac is just like an upper dilated structure that basically is continuous with the lacrimal duct. The duct is going to deliver tears to the nasal cavity on that, on whatever side of the face it's located on. And so when a person actually cries, what's happening is the tears that are rushing into the eye are going to be at a larger volume than can be drained. This is why they're going to start to overflow and stream onto your face. You also get a lot of drainage into the nasal cavity, and so that's why you tend to get a runny nose when you cry as well. All right, so let's now talk about the muscles that are associated with the eye. There are 6 extraocular muscles that control movement of the eyeball itself. Okay, so we have the superior rectus muscle. This is in the primary position of your eye, meaning looking straight ahead. The superior rectus muscle is going to aid in elevation. So you can see it's right there on top of the eye. So if you were to contract that muscle, of course, that would pull your eye back and up. Next is the lateral rectus muscle over here on the side. That is responsible for lateral movement of the eyeball, specifically abduction. Next is the medial rectus muscle. This works to keep the pupil at the midline. Inferior rectus muscle. This depresses, adducts, and helps extort the eye, which is a type of rotation. Then you have the superior oblique muscle, as well as that trochlea, which is the tendon that goes along with it. So the superior oblique muscle loops through this pulley-like structure, the trochlea, and it inserts into the sclera, which we'll discuss later, on the surface of the eyeball. So it's kind of like a pulley system. It gives the superior oblique muscle its action, so it can cause depression of the eyeball, even though it's inserted on the superior surface. Next is the inferior oblique muscle. Its actions are extortion, which is the primary action, but it can also elevate and abduct the eye. And then last, what you have is the levator palpebrae superior. So this is this green muscle shown in this image here. It does not move the eyeball, but it actually moves the upper eyelid, and so it can maintain the upper eyelid position. It's going to open. When it contracts, it's going to move the upper eyelid up, or sort of open your eye, as we would say. So next I want to talk about the layers of the eyeball. So starting with the fibrous tunic, this is the outermost layer of the eyeball. And it supports and protects the eye. The attachment site for the eye muscles is largely found on the fibrous tunic. It contains structures and it also assists in focusing. So the fibrous tunic consists of the sclera. The sclera is what we would say is the white of the eye. It's a very tough, dense, fibrous protective layer. It covers most of our ocular surface, and it's made of collagen and elastin fibers. And it also contains small blood vessels. The cornea is the other part of the fibrous tunic. This is a transparent front of the eye. It's much more fragile epithelial tissue, and it requires the protective tear film in particular. It is continuous with the sclera, so there's no specific dividing line, and that's shown here in this image. So it's a dense matrix of collagen, but they are organized because they can't interfere with light. So light is going to enter through this cornea, and if the collagen were not organized properly, a lot of that light would get diffracted before it would reach the retina. There are no blood vessels here, so the cornea receives its oxygen and nutrients pretty much only through the distribution of tears over the surface. One of the consequences of not having blood vessels is that, when you have damage, it does not have a much of an ability to repair itself. So any damage here can cause blindness, and to repair the cornea can sometimes cause or require a corneal transplant, again, because it has a limited ability to repair itself. The next tunic is the middle or vascular tunic. This tunic regulates light. Okay, it secretes and reabsorbs aqueous humor. The aqueous humor, if you look down here in this image, is basically the liquid that fills the inside of the eyeball. There's also some aqueous humor behind the cornea as well. So this circulates within the chamber of the eye. The middle tunic also controls the shape of the lens, and this is an essential part of focusing the light into a crisp image. So the middle tunic is going to include the iris, which is a muscle, okay, and that's shown here in the image. So the iris creates and adjusts the size of the pupil, which is shown there in the middle. So the iris is around, this is that pigmented part of our eye that gives us our eye color, and it surrounds the pupil. So it's going to dilate in response to dim light and constrict in response to bright light. And then, of course, it's pigmented, which is what gives us our eye color. Next is the ciliary muscle. So this is going to be on either side of the pupil and iris. The ciliary muscle and suspensory ligaments are going to hold the lens in place. Okay, and they will also adjust the lens to shape and focus the light at different distances. The lens itself is a clear protein structure. Like I said, it helps focus the light due to the action of the ciliary muscle and suspensory ligaments. Light that passes through the pupil will then pass through the lens. So the light's going to travel through the cornea, the pupil, and then it's going to hit the lens. The choroid is the blood vessel layer between the sclera and the retina. Okay, you can see this sort of bent over here in this image. It's covered by the sclera, so the sclera is outside of the choroid, and it attaches to the outermost layer of the retina. And so the retina, you can see here, is this yellow layer in this image. It delivers oxygens and nutrients to the retina, so it has an important role. Next is the innermost layer, the nervous or sensory tunic, and it's going to contain the retina. Okay, I've highlighted that down here. So the retina is the structure, or we've already discussed the retina, sorry, in the previous slide. It's the yellow area in this image. So it is the structure that contains the photoreceptor cells, and we're going to discuss the retina in depth in a few more slides. So the retina, again, is shown here in yellow. It contains rods, which are important for black and white vision, and cones, which are important for color vision. It also contains these receptors called IPRGC, and they respond to brightness levels as well as we think that they are important for establishing the 24-hour circadian rhythm. So the macula, which is shown here, has no rods. All right, but it has a high concentration of cones, and it's located where the vision or the visual will arrive after passing through the cornea and lens. Okay. So the center of the macula is called the fovea, fovea centralis, and this is located here in the back. And this is going to produce high acuity color bright light images. And so this is basically the area when the light passes through the cornea, the pupil, and the lens. It's going to be reflected onto this macula, or not reflected, but it's going to be directed to the macula on the back. And at the center of that macula is the fovea centralis. And that's where you have this high concentration of photoreceptors, so they are going to help you produce this high acuity, this sharp color image in bright light. There's also the optic disc. The optic disc is a location where the retinal neurons kind of fold back into the optic nerve, and because of this, it creates a tiny blind spot on the retina. And so we can't really see that due to the fact that our eye is constantly moving, and so that blind spot's always filled in with information from our brain. But there are ways to detect your blind spot if you're interested. Now, the remainder of the retina contain what are called rod cells, and so that's why our peripheral vision produces low acuity, black and white, low light images. And we're going to discuss the rods and cones as we move forward in this presentation. All right, so here we go, sectional view of the neural layer. So here we're looking at just like a slice of the edge of the eye. All right, this is a sectional view of that layer. And so what we can see are there are three layers within this. We have our superficial layer, which is located down here at the bottom of this image. This is where you have the retinal ganglion neurons that send axons to the optic nerve. You can see that those axons in blue down here at the bottom. And then if you look over here at this slide of actual tissue, that would be located at the bottom of this slide as well. Then you have the middle layer. The middle layer is going to include the bipolar neurons, which are colored pink in this drawing, as well as other connecting cells. And that is in this area of the tissue image. And then finally, the deepest layer, which is going to contain the photosensor cells, that's going to be the rods and the cones, are located here, and you can see them at the top of this tissue image as well. So let's discuss the rods and the cones a little bit. So how does our special sense of vision work, really? So what it comes down to are these photoreceptors. And the physiology of vision has to do with how these photoreceptors actually function. So the rods and the cones of the retina are what we would refer to as photoreceptors because they detect photons, okay? And so photons are just basic units of light. The rods are going to provide the central nervous system with information about the presence or absence of photons, but it's not going to give any regard to wavelength, okay? And so wavelength is what is going to be give us our perception of color. So the presence or absence of light, that is going to be determined and interpreted by the rods. The cones, they are going to be there to really assess what wavelengths of light are entering the back of the eye, hitting the retina, and that is going to give us our perception of color. So the names rods and cones, if you look in this image, is pretty clear that they have to do with the shape of the photoreceptor. So the purple one in this image is the cone, and this is going to give us our color perception, and then the rods are in beige. So both of these areas are going to contain pigments that are called visual pigments. Okay, so when light arrives, it's going to be absorbed in the outer segment of the photoreceptor, which I've just drawn up some arrows here. Okay, and so what these contain are these visual pigments. They have these flattened discs, and within these discs are the visual pigments, which is shown here on the right. That visual pigment is referred to as rhodopsin. It consists of an opsin as well as a retinal molecule. Okay, so the opsin is what actually detects color, and there are different opsins in different cone in the different cones. So then you have the inner segment here, and then what you have up here are these pigmented epithelial cells. Now they are going to capture any of the light that isn't actually, that doesn't interact with the rods or the cones. So next what we have here is this inner segment. The inner segment is just the part of the rods and cones that makes them just your general cell. It's going to take care of all the housekeeping functions. The nucleus is located there. And so this is just the parts of them that do your normal cellular functions with the added jobs of whatever types of specialized functions that these rods and cones have. At the very end, the opposite end of the outer segment, you have where the photoreceptor will synapse with a bipolar cell. And this is where it's going to send information, depending on light or dark or what color or what wavelength the rod or cone has interacted with. So I want to summarize this real quick. So light is going to enter the eye through the cornea, okay, here in red. Okay, and then the light will pass through the pupil, and of course that can be regulated by the iris. Okay, once light moves through the pupil, it's going to go through the lens, and that is going to focus it using the ciliary muscle and suspensory ligaments. Okay, and that's going to result in a focused image that's inverted on the retina. So then we will have our visual pathway come into play. All right, so in the visual pathway, we're going to have photoreceptors that are going to receive the light information or the wavelength information And then they are going to synapse that information to bipolar cells, which will then will synapse information with the ganglion cells. And then axons from those ganglion cells will proceed out of the eye, ultimately as the optic nerve. Okay, so here we have an image showing eyes at the top and the brain on the bottom. So these optic nerves that are going to come from each eye are going to proceed through the optic chiasm. And at this point, there's going to be an option, a little partial crossover. So you can see some of the nerves from one eye are crossing over and some of the nerves from the other eye. So this is mixing the information from the two eyes. Okay, so we have this combined visual field happening. The two optic nerves will then pass through the optic chiasm, and then ultimately they're going to travel to the visual cortex, where the image will be assembled. Now, other parts of the brain are also involved in sight, because you have parts of your brain that are going to decipher what you are seeing, and as well as why you are seeing it. So we're going to engage other brain areas for vision, but the actual part of producing that image is going to be in the visual cortex. And that is it for this presentation. Thank you.